This frog egg cell has been fertilized and starts dividing. The first cell divisions occur very rapidly. Cells divide every 30 minutes. This timing is very precise. Egg cells that have been fertilized at the same time divide and develop in almost perfect synchrony. After a day or two, embryonic development is completed, and tadpoles hatch from the eggs. Cells of this developing frog embryo rearrange in a dramatic ballet of orchestrated movements. In a continuous motion, cells from the outer layer of the embryo sweep towards one end, the vegetal pole, and start invaginating, forming a deep cavity in the interior. The paths of the cells and the topology of these rearrangements are best seen in this animation of an embryo that has been sliced open. The different cell layers that are formed in this way have very different fates. Cells that line the newly formed cavity, called the endoderm, develop into the lining of the gut and many internal organs such as liver, pancreas, and lung. Cells in the middle layer, called the mesoderm, give rise to muscle and connective tissue. Cells remaining on the outside, called the ectoderm, go on to form the outer layer of skin, as well as the nervous system. During development, a Drosophila embryo undergoes many complex morphological changes. We first see migration of pole cells from the posterior end. These cells are destined to become the germ cells of the fly. A crest develops, which separates a region that will develop into the head, mouth parts, and foregut. At this stage, the future tail end of the body is folded over on the dorsal side. Body segments then become defined. The first 3 segments will give rise to the head and mouth parts, the next 3 to the thorax, and the remaining ones to the abdomen. Eventually, the rear end of the embryo will retract back onto the ventral side and straighten out the embryo. Development to this stage takes about 10 hours. We can appreciate the complexity of these events by morphing a series of individual scanning electron micrographs into a continuous temporal sequence. Migration of pole cells. Development of various surface indentations, including openings to the air ducts or tracheal tubes. Segmentation, and tail retraction. A similar sequence viewed from the top, or dorsal side. Pole cells migrate, and then move into the interior as the hindgut invaginates. The rear end is temporarily folded over onto the dorsal side. And eventually starts retracting to straighten out the embryo. Early in development, when seen from the bottom, or ventral side, a deep groove forms during gastrulation, as mesodermal cells migrate inward, where they become the precursor cells for many internal organs. The groove then seals off as the cells that remain exterior zipper up.